Meiosis. Gametes. The sex cells of organisms are called gametes, eggs in females, sperm in males. In many eukaryotic organisms, the somatic cells, or non-sex cells, have two sets of chromosomes, which are called diploid. Gametes have one set of chromosomes, which are called haploid, and they are produced by meiosis. Sexual life cycles alternate between haploid and diploid phases. Fusion of haploid gametes during fertilization results in a diploid offspring. Meiosis. Meiosis reduces chromosome numbers in diploid organisms to create sex cells. Like mitosis, meiosis is begun by a single duplication of chromosomes. Unlike mitosis, the overall result of meiosis is 4 daughter cells, each with half the number of chromosomes, so they become haploid. Meiosis. The process involves 2 consecutive divisions, called Meiosis I and Meiosis II. Meiosis I finishes with 2 daughter cells, each with 23 chromosomes, so now they are haploid after that first round of meiosis. Meiosis II starts with the 2 haploid daughter cells and ends with 4 haploid cells through a process similar to mitosis. Meiosis One Summary. The nuclear envelope disappears and the spindle begins to form. Homologous chromosomes, replicated during interphase, pair to form a tetrad. Homologous chromosomes are matching pairs of chromosomes in somatic cells. Homologous chromosomes can carry different versions of the same gene, called alleles. Crossing over occurs. Genetic material is exchanged by the homologous chromosomes that were paired in tetrads, increasing the genetic variation of the offspring. Tetrads line up along the diameter of the cell. Their random order gives each cell a unique combination of genes from each parent's chromosomes, which is called independent assortment. Each tetrad splits. Maternal and paternal inherited chromosomes move to either pole, independent of the other chromosomes. Meiosis I Summary. The nuclear envelope reforms. The nucleus in each daughter cell is now haploid. The cytoplasm divides during interkinesis, similar to cytokinesis. The 2 cells are now haploid, but sister chromatids did not separate. Meiosis II Summary. Meiosis II follows the same steps as mitosis, separating sister chromatids. Meiosis II starts with the 2 haploid cells and finishes with 4 haploid cells. Karyotypes. Karyotypes are photomicrographs of stained metaphase chromosomes. At this phase of the cell cycle, chromosomes are condensed into characteristic shapes. We can look for large abnormalities and identify the sex of the individual. Non-disjunction events in meiosis I or II of a gamete can lead to abnormal chromosome number, as seen below. In the karyograph on the left, it is a normal karyotype for a human male. We know that because there's a Y chromosome here. In the karyotype on the right, we have Turner syndrome because we have one X female chromosome right here, but then there is neither an X nor a Y in the spot where there should be another chromosome for chromosome pair 23. Because there's no Y, this is a female, and it is a female with Turner syndrome.